Good afternoon, everyone. My name is Sangeetha Reddy, and I'm an assistant professor at UT Southwestern Medical Center, where I focus on identifying rational immunotherapy strategies in breast cancer. I lost my voice, so please bear with me as I get through this. Today, I'll be moderating the session on microbiome, diet, and fasting. There's a growing body of literature, due in part to those on the stage today, that the microbiome in the gut, in the blood, even in the tumor, plays an important role in shaping anti-tumor immunity and modulating responses to cancer therapy, including chemotherapy and also immunotherapies. Further, we now have an understanding that there's multiple mechanisms of modulating this microbiome through diet, fecal microbial transplants, as well as supplements to promote this immunity. We have 3 fantastic presentations today highlighting some of the advances in this field. I'd like to introduce you to our first speaker, Dr. Stephen Shao from Cedars-Sinai Medical Center. He'll be speaking on fungal regulation of breast tumor radiation response. Dr. Shao is an associate professor, director of basic science research in the Department of Radiation Oncology, and co-leader of the Experimental Therapeutics Program at Cedars-Sinai Medical Center. He received his MD and PhD in immunobiology from Yale University School of Medicine prior to completing his residency in radiation oncology at UCSF. Following his research fellowship with Lisa Cousins, he then joined the faculty at Cedars-Sinai Medical Center. The Shao Lab has a long-standing interest in understanding how the interaction between the immune system and the microbiome regulates the response to cytotoxic cancer therapy. In a recent publication in Cancer Cell, the Shao Lab demonstrated that the fungal microbiome regulates the anti-tumor immune response elicited by radiation. Dr. Shao?
Thank you so much for such a nice introduction, and I'd like to thank the organizers of the meeting for giving me the opportunity to speak here today. So as Dr. Reddy said, I'm going to speak a little bit about a different aspect of the microbiome today, and so hopefully by the end of this talk I'll have convinced you that there is more to the microbiome than just bacteria. So here are my disclosures. Okay, great. And so the genesis of this question really came from the conundrum that cytotoxic therapies like radiation and chemotherapy are actually immune events. And when we combine them with immunotherapies like anti-PD-1, in preclinical data, we've seen a lot of synergy. However, when we got to clinical data, as you can see from some of the trials like Pembro-RT or Neotrip, that it doesn't always work the way we think. And so, so even though we see it preclinically, we haven't seen it clinically. And so we wanted to really understand, so we know that cytotoxic therapies like radiation and some chemotherapies actually depend on their ability to induce an immune response. So their full efficacy is really because they can induce anti-tumor immunity. And so, like any other immune response, we really think that anti-tumor immunity mimics other immune responses like viral infections or things like that. And so we really are just asking the question, what governs the character and magnitude of this response? And so when we think about what governs an immune response, we think that immune responses are really shaped by the genetics of an individual, but also more importantly, the environment of the organism. And when we think of environment, we thought really long and hard about what that might be. And of course, right now, we're all thinking about how does the organism connect to the environment? And that's really through the microbiome. And the microbiome is actually a collection of all the organisms that are found in the human body. And you can see here from this very large cladogram that we found bacteria and other species throughout the entire human body. And so this is a little bit of a tiny background about why there's been such a renaissance in the microbiome. And so as you can see here from this beautiful picture, we've actually known about the microbiome for decades. But in fact, one of the reasons why it was so difficult to study is because you had to culture out the bacteria. And so for those of you who've done bacterial culture in medical school or graduate school or in any kind of research, you know that it's a very slow, laborious process. And so, which is why the microbiome was so difficult to study. However, the revolution that we've had over the last decade has really been in next-generation sequencing. And what you can see here is actually that when we talk about sequencing the bacteria, for the purposes of this talk, at least, and for a lot of these first phase of microbiome studies, we're actually really talking about looking at a very specific piece of the bacteria, namely this 16S ribosomal rRNA. And so, as many of you know, all bacteria contain 16S ribosomal RNA, and in order for us to be able to identify them, we're actually sequencing a small piece of this. And it can be quite variable, so everyone from V1 through V3, which are these variable regions in it. And what's really awesome about this is that the bacteria each are so unique that they are different in each of these variable species. So it's like each species contains their own barcode. And that's what it's enabled us to really identify not only a wide diversity of things, but things that are not culturable. And so that's really dependent on our knowledge of the database as well. So the database is just as important as the sequencing. So how we identify them, so you can have a barcode, but if you don't know what the barcode is, you can't identify it. And this gets to be important later when we start talking about fungi. So hold on. And so the beginning of this, we really recognized from our colleagues, some of whom are going to be speaking in a few minutes, that the bacteria are really important for both chemotherapy and immunotherapy. And there were a number of different mechanisms that were identified, but we decided to ask just the basic question, does the microbiome also regulate the tumor response to radiation? And perhaps not surprisingly, so what we did is we did a mouse model in which we took C57 Black 6 mice, injected them with a syngeneic mammary carcinoma, EO771, which mimics triple-negative breast cancer, and then we depleted them with antibiotics. So antibiotics in this context is very different than antibiotics we give to patients, for instance. And this is a very depleting one meant to cover, it's broad spectrum, meant to cover all the bacteria that we know of. So this is a quadruple regimen that's given to mice over a long period of time. So we deplete the bacteria and then we irradiated them. And so perhaps not so surprisingly, given the chemotherapy and immunotherapy data, what you can see here is that the control in the black and the green is the radiated ones, and that radiation slows tumor growth, as we would expect. But that when you have antibiotics on board, that the radiation no longer has any effect on the tumor growth. And that's mirrored in the survival, in which when mice are depleted of their bacteria, the response to radiation is significantly attenuated. And when we looked at the tumors themselves and we evaluated their cell division through BRDU, you could see that the bacteria actually doesn't allow radiation to inhibit the cell division, and it also curtails the cell death that's caused by radiation. So supporting the notion that it really, without the bacteria, that tumor growth is significantly less sensitive to radiation. And so one of the things, of course, you do when you study the bacteria is you just want to check to be sure that the bacteria is gone. So we did a qPCR to look at it. So we look at 16S, which is the ribosome, and to see. And then one of the things that was really surprising, though, so of course we saw that there was a depletion of the bacteria, but we were looking for fungi as well as a control, and we saw that, in fact, when you look at for fungi, that the fungi go up dramatically. So we were surprised. In many cases, it was thousands of fold more than baseline. And so we went to see, well, what about the fungi? So when you look at this and you think about it, all of the previous work that I just described was really about the bacteria. And so when people think of microbiome, they think of the bacterial microbiome. So what about the fungi? And so you might ask, why do people not study fungi before? And as this pie chart shows, there are very, very few fungi. So you can, if you could really look really closely, you'd see that there's a small red sliver that represents the number of fungi in the stool. However, so you can see that it's barely visible. But interestingly, fungi, because they're eukaryotes, are actually 100 times larger than bacteria. So if you calculate it by biomass, in fact, the fungi are a meaningful but still small portion of the microbiota. And so, lest you think that we were just making this up, many people have also studied the fungi in the context of stool. And so if you look out in the wild, so there's this a great study in Science where they looked at wildling mice, so mice that are pet store mice or mice from the fields. And you can see that, in fact, they contained significant amounts of fungi. And we believe that in the human, kind of out in the wild as we are, we also have a significant amount of fungi. And also, a recent work from our colleagues Ravid Straussman in Israel and Ilyan Ilyev at Cornell have shown that not only are there fungi in the stool, but there are also fungi in breast tumors themselves. And so they were able to identify this across a number of large cohorts. So fungi, though, are a little bit different than bacteria. So this is why I explained a little bit about why we do the bacteria. But fungi, because they're eukaryotes, don't have that exact same barcoding. So in fact, when we look at their species, we have to look at a different part of the ribosome called the internal transcribed spacer, or ITS. And this allows us to look at the fungi, but we also look at the 18S ribosome, and we have to look at a different portion. And the databases themselves are not as well curated. So though our work itself was done from an in-house database that we curated through much labor of blasting of yeast species. And so, but nevertheless, we went back and looked into see what happens when you deplete the fungi. And so we went back and asked that same question where we treated mice with antifungals and then irradiated them or gave them antifungals and alone. And we saw that, in fact, here you can see that, again, radiation slows down tumor growth. But what surprised us was that the combination of fungal depletion plus radiation dramatically slowed tumor growth and improved survival. And when we looked at the tumor cell division and tumor cell death, we found that, in fact, it didn't really affect the ability of radiation to slow down tumor division, but it did significantly increase tumor cell death. And of course, when we looked at them by amount, we could see that it doesn't really affect the bacterial level, but it did affect the fungal levels. And so then when we look back at the immune component, we saw that antifungals increased the immune activation following radiation. So we saw that when you looked at the activated CD8 positive T cells or CD4 positive T cells, that in fact bacteria, when you deplete the bacteria, there's significantly less activation, but when you have antifungals, there's significantly more. But perhaps more interestingly is that antifungals also simultaneously decreased immunosuppression, and this was a very dramatic effect that we saw when you look at the PD-1 positive, CD8 positive T cells, as well as the CD206 F480 positive macrophages. You can see that in these red dots that both of these are significantly reduced compared to either radiation or radiation plus bacterial depletion. And so no microbiome study would be complete without looking at the bacteria and fungi themselves. And so we sequence them, of course, using 16S and ITS sequencing. And so this is at the family level, and you see this stacked bar. And I think what's pretty dramatic here, so there are a number of changes that you can see, but in the red, you can see that with antibiotics in the fungi, you can see an almost complete kind of overgrowth of the Saccharomyces family. And interestingly, when you use this technique called LEFC, which is basically a statistical technique to identify which species are attributable to this effect, you can see that familiar names like Saccharomyces and Cerevisiae come up when you look at what is the reason, which fungi are responsible for the changes following antibiotics. And when we look at this more closely and we sequenced out the Saccharomyces in Candida, we found that indeed you see an overgrowth of members of the Saccharomyces family, which include Saccharomyces cerevisiae and Candida albicans. And so we went on to prove in a number of gnotobiotic experiments, but one that I think, this one is the one I think is the most beautiful. We used mice in which we created fungal-free mice. And so we colonized mice with just bacteria, so a defined colony of bacteria called the alter shade-lord flora, or ASF. And then we treated them with radiation or not. And what you can see is similar to fungal depletion with antifungals, that when there was no fungi present, that radiation is significantly improved, and that it mirrors what we see immunologically as well, in which there's a decreased amount of immunosuppression leading to more activated T cells. And so finally went to look at patients. And so in order for us to be able to look at patients, we wanted to understand a little bit about how patients would be seeing fungi. And so we looked at the upstream receptor of C-type lectin receptor called dectin-1, which is the main sensor of fungal cell walls, or beta-glucan. And first we looked in human breast tumors and we asked, can you see dectin-1 positive cells? And in fact, you saw that in triple-negative breast cohort that you can see that there are a number of dectin-1 positive cells. In fact, in some cases, it represented 20 or 30% of the cells. And then when we went back to the TCGA and looked at dectin-1 expression and normalized it against the number of CD11b or C positive cells, you found that of those patients that had high expression, that they had significantly worse survival compared to those that have low dectin-1 expression. So in conclusion, we found that depleting the bacteria significantly reduces the effect of radiation on tumor growth by curtailing the anti-tumor immune responses, but that antibiotic treatment leads to overgrowth of fungal species, and that targeting fungi enhances the response to radiation, and that fungal depletion enhances the efficacy of radiation by increasing the number of activated cytotoxic T cells while simultaneously decreasing the immune cells, the immunosuppressive immune cells, through a mechanism mediated by dectin-1. And so we think that, as we kind of work through this question, that there is a counterbalancing force. So we don't deny that the bacteria are very important for the generation of anti-tumor immunity following cytotoxic therapy. We think that they're really critical and they provide critical inflammatory stimuli. However, like we've seen with PD-1, that the immunosuppression may be equally or more important in that, and we believe that that immunosuppressive capacity is actually regulated by sensing alternative organisms in the microbiome, such as fungi. And so just to kind of give you a teaser about what's coming up, we also decided to ask, not only is it important in radiation, but is it also important in immunotherapy? And we found that, in fact, similarly to radiation, that in this model using anti-PD-1, that you can interestingly convert a model that doesn't respond to anti-PD-1 into one that does. And so when you deplete the fungi. So we're very excited about this kind of new avenue, and we hope that it will have profound implications for the future. So thank you very much.
Please come to the microphone with any questions.
Oh.
I'm going to start with one, if you can hear me. Given that we know the composition of the microbiota, of the bacterial microbiota impacts response to immunotherapy, how do you know in your last couple experiments whether the antifungal was due to changing the composition of the bacteria or truly due to fungal depletion? And even that in that ASF experiment, how do we know it's not that specific microbiota compared to wild type that may have contributed?
That's really a great question, and in fact, we haven't quite teased that out, that in fact, you do see a fair number of changes in the bacteria when fungi aren't present. And so, it may be that there's an increase in the activation, but we have done a number of monocolonization experiments where we've given only fungi or only bacteria, and you can see that fungi do seem to have a pretty profound effect on the immunosuppressive capacity, but generally leave the cytotoxic kind of aspect alone. So.
It's a really interesting question. So you kind of hinted at it in your model already, because I was going to ask, is it that the bacteria are increasing the response or the fungi are holding it down? Of course, it could be both. So have you done the experiment where you clear them both?
Yes, so we've done one where we cleared both, and we've also done it in germ-free mice that have neither. And what you find is that, in fact, probably the bacteria are dominant. And so I think that you absolutely have to have the bacteria to generate an appropriate cytotoxic T cell response. So I think that the fungi, and if that overwhelms the suppressive arm of the fungi, I think it's not allowed to be. You don't get an appropriate response, so there's nothing to suppress from the fungi. So doing both kind of, it gets overwhelmed by the bacterial effect.
Hi, Trent Megenstock, Beatrice. What antifungal agent are you using in these?
So in the data I presented, we actually used fluconazole, but we've actually tested it in multi-agent antifungals where we used amphotericin as well as 5-FC. kind of one of the secrets about, I guess, in the fungal world. So as you know, there are only a few pathogenic fungi, and so a lot of the antifungals are directed specifically at those species. So I don't know if we cover all the fungi the way we do with the bacteria. So.
Sven Becker from Frankfurt. Thank you very much. This is pretty crazy stuff. As you say, the microbiome, we associate that with the skin and the gut. But you said at some point that there are fungi inside of breast tumors. How come my pathologist has never told me about this? Is it rare? Are they there all the time? Do they see them? And of course, the next question would be, has anyone looked at the implication of, fungi in the tumor with regard to prognosis? Thank you.
Right. So one of the reasons is it is true that they're rare. So they're not florid like infections in some ways. You have to look for them pretty carefully. How they identified them in the 2 parallel cell papers that just came out was really through a combination of deep sequencing as well as fish-based techniques and some antibody-based techniques, and they're still quite rare, so not all breast tumors have them, in fairness. I think it does relate a little bit to how they're connected to the environment. So especially for breast tumors, the larger they are, the more necrotic they are, and the more ductal involvement may have some role in that. But if you look at some of the ductal lavage data, which I know is a little bit kind of out there already, you can see that they can find bacteria, and we speculate that there are also fungi in the ducts themselves.
Unfortunately, we'll need to move to our next presentation, but I'm sure Dr. Shao would be happy to answer any questions afterwards.
Yes, thank you.
Thank you, Dr. Shao. Our next presenter is Dr. Guido Kromer from University of Paris, who will be speaking on fasting and microbiotic. Dr. Kromer is a professor at the Faculty of Medicine of the University of Paris, director of the research team Metabolism, Cancer, and Immunity at the French Medical Research Council, director of the Metabolomics and Cell Biology platforms of the Gustave Roussy Comprehensive Cancer Center, and hospital practitioner at the Hospital European Georges Pompidou, Paris. Dr. Kromer's work focuses on the pathophysiological implications of cell stress and death in the context of aging, cancer, and inflammation. He discovered the ignition of regulated cell death pathways by mitochondrial membrane permeabilization, the cytoprotective and anti-aging effects of macroautophagy, as well as the decisive role of immunogenic cell death in anti-cancer treatments, and is currently working on microbiome and determinant summit. His contributions have been recognized with multiple awards, including the most prestigious cancer research prizes from Belgium, France, Switzerland, Italy, and the European Union-sponsored Descartes Prize. Thank you, Dr. Kromer. Please come up.
Thank you. So I will talk mostly about dietary influence on breast cancer and briefly touch up on the microbiota. So everybody knows here that an unhealthy lifestyle, sedentary, and poor diet is a major risk factor of breast cancer as well as for other cancers. And that aging is, of course, the most important risk factor for most kinds of cancer, including breast cancer, through effects that affect inflammatory and immune-relevant microenvironmental factors. So this is an example of a mouse experiment in which breast cancer is induced by continuous progesterone receptor stimulation together with DNA damage. You see that wild-type mice develop in this protocol breast cancer with a latency of several weeks or months, and that an immune defect caused by the lack of the pattern recognition receptor FPR1, formyl peptide receptor 1, grossly accelerates the development of disease as a proof for natural immunosurveillance in breast cancer carcinogenesis. And if we move to humans, it turns out that there's a frequent loss of function polymorphism in FPR1 that affects 34% of humankind, either in heterozygosity or in homozygosity. And if you look at the pan-cancer analysis of the TCGA, it turns out that cancers develop 9 months earlier in patients that have a heterozygous loss of function mutation in FPR1, and 2 years earlier in patients that bear this defect in homozygosity. And you can add up to 580 million years of cancer-free life that is lost for the current humanity of 8 billion inhabitants, meaning that this defect in FPR1 is quantitatively the most important genetic defect precipitating the diagnosis of malignant disease. And if you look at the luminal B breast cancer subtype, patients who are heterozygous or homozygous in the FPR1 mutation, which is more than 1/3 of women across different continents, develop breast cancer as an average more than 5 years earlier than patients bearing the normal alleles. And this can be validated in the French cohort, the STORM cohort, in which we genotyped a little bit more than 200 women with luminal B breast cancer, in which the loss of function of FPR1 accelerates breast cancer diagnosis by more than 6 years. And this difference in age is also found for the age at metastasis and the age of death, meaning that FPR1 itself has no impact on the prognosis of breast cancer, but has a major impact on diagnosis, and would be interesting to incorporate this information in future interception and cancer prevention trials. Now, there are additional arguments in favor of immunosurveillance of hormone-induced breast cancer of the luminal B type that we characterize here in mice. So, for instance, the severe immunodeficiency caused by the RAC2 interleukin-2 receptor gamma knockout accelerates the development of breast cancer and reduces overall survival in this mouse model. And there are other arguments. For instance, if we look at the effect of tamoxifen, which of course delays the induction of progesterone-induced breast cancer in mice, this effect is found immunocompetent but not in immunodeficient mice, suggesting that hormone therapy in breast cancer, and as we have recently shown for prostate cancer, is at least partially mediated by a modulation of the immune system in favor of immunosurveillance. So we did a systematic analysis of different immune defects that might accelerate or decelerate the development of hormone-induced breast cancer. You can see here the usual suspects that were characterized with respect to the incidence of breast cancer. And the global analysis of these results suggests that defects in the interleukin-21 system, in the interleukin-22, NKG2D, and the two interferon systems, type 1 interferon and interferon gamma, accelerate breast cancer, while defects in inflammatory signal postpone the diagnosis for breast cancer in this mouse model. And if we look at the time from diagnosis to death, which in women would correspond to prognosis, it turns out that the knockout of IFNA, of interferon gamma, or the depletion of CD4 positive T cells are particularly dismal in their effects on the development, the progression of the disease. Now, with respect to aging, we will publish at the beginning of the year an update of the hallmarks of aging in Cell. And in these hallmarks, we included dysbiosis as a cardinal feature of the aging process, which is, of course, also a hallmark of cancer. And so this is interesting to look at the parallel alterations in aging and cancer that affect the microbiota, a reduction in diversity, a depletion of beneficial microbes, and an elevation in the incidence of harmful microbiota, which are, of course, related mechanistically to obesity, diabetes, and cancer, because cancer itself can change the microbiota and cause dysbiosis that resembles that found in aging. And this is actually due to the fact that in mice that even be relatively small tumors that are close to undetectable And in patients, according to literature, cancer can induce an elevation of the adrenergic tonus, stimulate beta-adrenergic receptors, and then change the microanatomy of the ileum. So it is causing an ileopathy that at the end causes a cancer-related dysbiosis. So we did a meta-analysis of cancer-related changes in the microbiota that we call GOMS, gut oncomicrobiome signatures. And you see that across different cancer types, including breast cancer, there are very similar changes leading to an up or overrepresentation of harmful bacteria in the feces of patients, or a depletion of useful bacteria, including, for instance, ruminococci that Jennifer Vargo will talk about later on, I suppose. So we did a systematic analysis of nutritional effects, for which we suspect, of course, that part of them will be mediated by the microbiota, but certainly most of the effects are mediated also by the immune system. So we performed different alterations of the general diet, so high-fat diet, which accelerates breast cancer development in mice, or intermittent fasting that causes a delay in the breast cancer diagnosis and actually improves prognosis. The way how this experiment is done is it is an intermittent alternative day fasting protocol, 1 day on, 1 day off, without any long-term changes in the weight of the mice, but an impressive delay in the development of breast cancer. And you can see that we see also effects of micronutrients on the development of cancer. So vitamin B3, calcitriol, vitamin B6, pyridoxine, and nicotinamide, vitamin B3, all given at pharmacological doses, not at doses that are supposed to compensate for hypovitaminosis, are able to postpone the development of breast cancer. And these effects are interesting with respect to the effects of caloric restriction or intermittent fasting. We know that these involve a reduced activation of nutrient sensors, an enhanced activation of nutrient scarcity sensors, with the final outcome of the induction of autophagy and other stress responses in cancer cells and immune cells, the formation of ketone bodies that have themselves anti-inflammatory and immunostimulatory factors, also useful in a combination with immune checkpoint blockers, and pro-immune cell stress responses. And if we more specifically remove proteins from the diet of mice, or we use plant-based only diets that lead often to a methionine restriction, we obtain a compensatory synthesis of polyamines that rejuvenate the immune system, stimulate autophagy in cancer cells, and activate anti-cancer immunosurveillance. If we deplete carbohydrates, which is particularly useful in the current state of the American diet, we can expect an improvement of the intestinal barrier function, a reduction in the metabolic support of cancer, and a reduction in comorbidities that affect the prognosis of cancer patients. And with respect to lipids, of course, there is a literature suggesting that low-fat diet has a positive impact on the incidence of breast cancer, but there is also a literature suggesting that ketogenic diets, in which most of the calories are consisting in lipids, proteins are just dosed at the minimum amount, and carbohydrates are severely depleted, lead also to the formation of ketone bodies, exactly like starvation, with their relief of immunosuppression anti-inflammatory effects, reduction of glucose supply to cancer cells, and globally positive effects. So let us move to the micronutrients that we characterized in our system. Pyridoxine, which is vitamin B6, delays the manifestation and increases the lifespan of mice in this progesterone-induced breast cancer model, only in immunocompetent, not in immunodeficient mice. It's again an immune-dependent effect. And similar findings were obtained for vitamin D3, calcitriol, that I remind you has been used in clinical trials to suppress the development of ductal carcinoma in situ in women. And again, the effects of nicotinamide, a precursor of vitamin B3, that are quite spectacular at all levels, tumor-free survival, overall survival, and prognosis can found in different models of breast cancer, are depending on the immune system because depletion of CD4 and CD8 positive T cells or neutralization of interferon gamma with antibodies abolishes the positive impact of nicotinamide on breast cancer development. And nicotinamide can actually be used to treat established cancers in mice with curative effects, tumor growth reduction and increased overall survival that again depend on T cells and can be actually amplified by PD-1 blockade, just as another proof that this system is working through the activation of the immune system. And additional results in favor of this hypothesis can be obtained by high-dimensional cytometry, showing that interferon gamma or TNF, or both together, are overproduced by tumor-infiltrating CD4 and CD8 positive T cells in the tumor infiltrate of mice bearing cancers treated with nicotinamide. And the single-cell sequencing confirms this data at the RNA level, showing that, for instance, interferon gamma is produced at higher levels by the tumors that are developing in the context of nicotinamide treatment. And you also see here an enhancement of type 1 interferon responses within the tumor, and neutralization of type 1 interferon or interferon gamma indeed abolishes the therapeutic efficacy of nicotinamide against established cancers. So we have some data on the mode of action of nicotinamide, which is a precursor of NAD, which is a cofactor of enzymes such as sirtuin-1, PARP1, and many others. And through this modulation of enzymatic effects, we obtain a reduction of T cell exhaustion, an improvement of effector CD4 and CD8 T cell functions, and also an alteration of the myeloid lineage in favor of improved anti-tumor functions of M1 macrophages. All this together improves cancer immunosurveillance. So in conclusion, age is the most important risk factor, and age-decelerating lifestyles, of course, reduce breast cancer risk. Breast cancer is under strong immunosurveillance, exemplified by the loss-of-function mutations of FBR1. Breast cancer patients exhibit gut oncomicrobiome signatures, or GOMS, that resemble those found in other cancers. Intermittent starvation, fasting, and several micronutrients, including the vitamin D3, vitamin B3, and vitamin B6, improve breast cancer immunosurveillance via a variety of mechanisms that include caloric restriction like or mimetic effects. And among these agents, nicotinamide, which already has been shown in the past in a phase 3 trial to prevent non-melanoma skin cancer, emerges as a potential anti-aging and anti-breast cancer medication that could be tested in window of opportunity trials. And with this, I mention my acknowledgements, and I thank you for your attention.
Thank you so much for that wonderful presentation. I think we have time for just probably 2 questions. We'll take one online and we'll take you in just a second, in person. So the first question is from Dr. Preeti John, who's asking, do we know if weight loss medications can help with reducing that breast cancer risk reduction associated with the variables you were talking about?
Yeah, we are indeed in a period in which new weight loss medications are popping up, the so-called GLP-1 agonists that show clinical utility, at least in clinical trials, although it remains to be determined whether they will be successful in the real world in spite of their major side effects. And of course, it would be expected that long-term weight reduction, even if it is achieved not by diet but by medication, would have a positive impact on breast cancer development and breast cancer prognosis.
Our final question.
Maybe you move to the microphone that is working.
Okay, Nise Yamaguchi from Brazil. Dr. Shao, I think that we might take advantage of the information that we have that low dose oxygen is also impairing the activity of radiation therapy. And there are trials with hyperbaric oxygen and also ozone therapy that might improve the activity of radiation. Do you think that might work also in the gut microbiome like fungal infections? Because we see from what we have in nature that when we don't have so much oxygen, the fungi, they can grow more.
To hijack that, but I guess I'll just answer briefly. I think that interestingly, I'm not sure what effect it would have on the gut for hyperbaric. It might, but I suspect it might actually have some effect on the intratumoral microbiome. But I guess that would be something to be seen.
And to Dr.
We may have to stop there so we have time for the next presentation.
Okay.
Thank you so much, though, but please come back afterwards. Thank you, Dr. Cromer, again for that fantastic talk. Our final presenter will be Dr. Jennifer Wargo, my prior mentor from MD Anderson Cancer Center, who will be speaking on microbiome in response to immunotherapy. Dr. Jen Wargo is currently a professor of surgical oncology and genomic medicine and the leader of the platform for innovative microbiome and translational research at MD Anderson. She completed surgical residency at the Massachusetts General Hospital, followed by 2 fellowships in surgical oncology with a focus on cancer immunotherapy. She was recruited to MGH in 2008 to join the faculty in the Division of Surgical Oncology and established a translational research laboratory focused on better understanding response and resistance to treatment for melanoma, pancreatic cancer, and other cancers. During that time, her laboratory demonstrated that molecularly targeted therapy could sensitize tumor cells to immunotherapy, providing the rationale for combination therapy. She also began studies on the tumor microbiome in pancreatic and other cancers while at Harvard. Dr. Wargo was recruited to MD Anderson Cancer Center in 2013 to lead the melanoma moonshot efforts and translational research work on targeted therapy, immunotherapy, and the impact of the gut and tumor microbiome in cancer. Dr. Wargo.
Thank you, Dr. Reddy. Thank all of you for being here today and welcoming a melanoma surgeon to your breast cancer conference. I'll try and do this justice. So today I wanted to talk about the microbiome in response to immunotherapy. Let's see. This is not advancing for some reason. Yeah, it did.
We're having trouble advancing the slides.
Can you advance the
Not
slide?
advancing.
Oh, there we go. Okay. All right. So notably, I do have a few disclosures. Sorry, guys. Can you help out? That's what I'm doing. Okay, here we go. All right. Anyway, so before I get started into the actual meat of the talk, if you will, although it doesn't include much meat because I've now gone plant-based, but I wanted to thank everyone who's contributed to all this work, and that includes patients and their families, all care providers, the Melanoma Moonshot team at MD Anderson, Prime TR, this program for innovative microbiome and translational research. MD Anderson Cancer Center is an amazing place. The leadership is great, but also supporters and collaborators worldwide, including Guido and Laurence Vogel and others. And thank you to everyone really worldwide who's contributing to advance cancer care through science. And so So really, as a background, I wanted to highlight that we've made major advances in cancer treatment with the use of immunotherapy and other approaches. Immunotherapy has made its way as one of the very pillars of cancer care. We've actually seen an overall reduction in cancer-related mortality, which is brilliant. But what I would argue is that there's still a critical need to improve responses to cancer therapy and to limit toxicity in patients with established cancer, as well as opportunities to prevent cancer altogether. And so through translational research, we've identified a number of factors that impact tumor growth and immunity that can be targeted. And this includes host intrinsic factors like the tumor and the tumor microenvironment, also host genomics, systemic immunity, systemic factors like sex hormones, and then also the microbes that are within us. But it also includes factors like Dr. Cromer spoke about, which are host extrinsic, or the so-called exposome. And this includes things like environmental exposure, psychosocial factors like stress, lifestyle, like diet, critically important, and then also the microbes that surround us. And considering that humans and organisms represent a complex ecosystem, what is the role of microbes that coexist with us on cancer and on therapy response? And this will be a bit of a broader overview and will help address one of the questions that was asked earlier. So here's a great cocktail party tidbit. Okay, so 3.5 billion years ago, microbes actually helped shape the earth for future forms of life. They actually gave us the very oxygen that we breathe today. And in fact, if you want another cocktail party tidbit, there are more microbial genes in the human body than there are stars in the galaxy. Okay, so feel free to use that one. It's a great one. And now we know that 3.5 billion years later, it's very clear that microbes are actually pervasive in our environment and actually within living organisms. Now take a human, right? There's 20,000 human genes, which represents 1% of the total genomic content. But in that same human, there's between 2 and 20 million microbial genes, right? And that represents 99% of the total genomic content. And importantly, these are inherently modifiable. And advances in next generation sequencing have really allowed us to better understand these microbes. And so the importance of the microbiome in health and disease is increasingly evident, and this includes in cancer, where microbes within the tumor and the surrounding tissues, as well as microbes within the gut and other niches, can actually impact carcinogenesis, therapy response, and other physiologic processes. And in fact, these polymorphic microbes made their way to the hallmarks of cancer and the enabling characteristics, which was recently updated by Doug Hanahan, published in Cancer Discovery just a few months ago. And I really posed the question, could microbiome targeting become the next pillar of cancer care with strategies to monitor and modulate the microbiome, to treat, intercept, and maybe even prevent cancer someday? And really a provocative question here. And so we have a growing understanding of how microbes in the gut can actually impact immunity and response to cancer treatment. And if you think about it, all along this long alimentary tract, on one side you have this dense network of microbes, and on the other side you have this infiltrating network of immune cells. And so there's this constant to and fro and dynamic interaction all along that vast surface area. And it goes without saying that these microbes could actually profoundly influence our immunity. And early studies supported a role for gut microbes in shaping response to therapy, with validation in numerous clinical cohorts of patients on immune checkpoint blockade. So interestingly, some of the earliest work was done by stem cell transplanters who studied patients with AML, and this was out of Memorial Sloan Kettering Cancer Center. And they found that if patients had a more diverse gut microbiome, which is generally associated with better overall gut health, that they actually had much better outcomes on treatment with stem cell transplant for AML. Whereas if they had a low diversity of gut microbes, they actually did very poorly. Now, in addition to this, brilliant investigators, including Laurent Zedvogel and Tom Gajewski, showed that in preclinical models of treatment with immune checkpoint blockade for cancer, that depending on what the bugs look like in the gut of mice, dictated whether or not they responded to treatment with immunotherapy, and furthermore showed that you could actually change the microbiome and make these mice respond better. And so what I can tell you is that we had been studying the tumor microbiome, but saw this data presented at the Society for the Immunotherapy of Cancer by Tom Gajewski, absolutely floored by these results, and so asked the question, had they studied this in patients, and they hadn't. So we saw this as a unique opportunity to do that. And so at MD Anderson, what we did is actually wrote a protocol to collect oral and gut microbiome samples, fecal samples, in patients with metastatic melanoma going on to treatment with immune checkpoint blockade. And how did we do this? It was a clinic-based collection, so we would swab their cheeks in clinic, 100% compliance there. We would give patients a kit so they could take it home, and in the privacy of their own home, they could collect their fecal microbiome sample or gut microbiome sample, and around 50% of patients mailed those samples back. But I can tell you people are very excited to mail back their gut microbiome sample now. And what we found when we did this is we sequenced the gut microbiome, and we found that just like the stem cell transplanters found, if patients who responded to treatment with anti-PD-1 had a more diverse gut microbiome, okay, suggesting that diversity matters and that high diversity is good. And they had prolonged progression-free survival compared to those with a low diversity. In addition to this, we found that different bugs in the gut were found in responders to therapy, namely things like Faecalibacter, Rhumatococcus, Clostridialis, whereas patients who did not respond to treatment with anti-PD-1 had a higher abundance of different bugs like Bacteroidetes. And in fact, we worked with Laurent Sitvogel and Tom Gajewski to co-publish 3 back-to-back papers in Science in 2018, which really opened up the field, suggesting that gut microbes could indeed be predictive of response to immune checkpoint blockade in patients with cancer. And we now know that gut microbes can also modulate the tumor microenvironment in patients with breast cancer, and this is a recent paper just on this, which provides opportunities to target gut microbes to improve outcomes. And so, hot off the press, if you will, in cancer immunology research, reciprocal interactions between the gut microbiome and mammary tissue mast cells promote metastatic dissemination of hormone receptor positive breast cancers. And you can see every day there's a new publication on the role of gut and tumor microbes. And I posed the question, based on all this, can we actually modulate the gut microbiome to enhance response to immunotherapy and or to abrogate toxicity? And I used to just say this optimistically, but now we have cold hard data to suggest that this is indeed the case. And so efforts to target gut microbes to improve response to cancer treatment can be effective. And just a few years after those seminal papers were published in Science, investigators actually ran clinical trials in patients with melanoma that had progressed on anti-PD-1 and other forms of immunotherapy, and showed that if you take those patients and wipe out their inherent microbiome, give them a fecal transplant from a responding patient, either a complete responder or partial responder, you can actually get reversal of immunotherapy resistance in those patients. Really dramatic, okay? And back-to-back papers published in Science just a few years ago showing just that. This was from a group from Israel, showed in 10 patients treated, they had a 30% response rate, which in PD-1 refractory metastatic melanoma is pretty darn good. And a parallel paper published back-to-back out of University of Pittsburgh showing that clinical response is seen in, I believe, 6 out of 15 patients with a favorable change in the microbiome with a higher abundance of Rhumatococcus and higher abundance of other good bugs like Faecalibacter and Clostridialis, and also a change in serum metabolites. Now, does it take a full fecal transplant? Not necessarily so, and others who have studied kidney cancer have actually showed that if you take patients who were being treated with combined checkpoint blockade, targeting CTLA-4 and PD-1, if you actually add a commonly prescribed probiotic in Japan, the CBM 588, which is a Clostridium butyricum preparation, you could actually significantly improve outcomes in those patients, not only from progression-free survival, but also overall survival. But these data need to be replicated. Nonetheless, it suggests that not only can a fecal transplant work, but a targeted bacterial intervention in the gut can actually be associated with improved outcomes to cancer immunotherapy treatment. And importantly, there is crosstalk between the gut and tumor microbiome, which substantiates the rationale for fecal transplant and other microbiome modulation strategies in other cancers. And so at MD Anderson, Florencia McAllister, she treats patients with pancreatic cancer. She studied long-term versus short-term survivors of patients with pancreatic cancer and found that long-term survivors actually had a more diverse tumor microbiome. And that she then studied and found that a lot of the bugs that were present in the tumor could potentially be derived from the gut. Now, she next did some preclinical studies where she took mice, implanted tumors, pancreatic cancer into the mice and then gave these mice fecal transplant from either a long-term versus a short-term survivor versus a healthy donor, and found that if she gave fecal transplant from a long-term survivor, they actually had delayed tumor outgrowth and a brilliant immune response within the tumor. And she actually now has a clinical trial underway in which she's treating patients with pancreatic cancer in the pre-surgical setting with a fecal transplant from either a long-term survivor versus a healthy donor. Pretty phenomenal studies. In addition to this, other investigators, including Mike Overman at MD Anderson, is studying the use of fecal transplant in reintroduction of anti-PD-1 in patients with MSI-high cancers. Started off with colorectal cancer, but he's accruing other cancer types as well that are MSI-high that are refractory to treatment with anti-PD-1. He's actually seeing clinical responses in this, where he actually takes those patients, wipes out their inherent gut microbiome, transplants in fecal transplant from a responding patient, and has seen pretty phenomenal responses in that trial. And studies of the intratumoral microbiome in breast cancer also suggest that, that microbes are pervasive. Now, so we had actually started to study the tumor microbiome many years ago, and I studied this with Ravid Straussman when he was still at Harvard. He's now at the Weizmann Institute, and found that in patients with pancreatic cancer, you could actually identify microbes within the tumor that were actually breaking down chemotherapy into its inactive form. But is this happening in breast cancer? And so Ravid has done brilliant work. He recently published a paper just a few years ago in Science where he showed pan-cancer, you can actually identify bacteria within the tumors of these patients, right? And could actually find that distinct microbial signatures are associated with different subtypes of breast cancer. In addition to this, it's not just about the bacteria, right? You can identify viruses, you can identify fungi, you can even identify parasites within tumors, right? Who would have thought that, everyone thought tumors were sterile for many years. Well, guess what? They're not. And advances in next generation sequencing have allowed us to understand what microbes are there and what they might be doing. But there's a lot of complexities with it with regard to potential contaminants. And so I think when you look at the tumor microbiome, you have to take things with a grain of salt. There's a lot of validation that needs to be done. But nonetheless, there's brilliant work being done. The role of the tumor microbiome is really critical, and the role of the gut microbiome is as well. But a lot of considerations really exist as we use approaches to modulate both the gut and the tumor microbiome in patients with cancer. And why is this? Well, Jen McQuaid and I put out this paper in Lancet Oncology a few years ago. And when you think about it, this is a living drug that we're giving patients, right? So before treatment, we have to consider what patient population do we study? Should the microbiome be profiled? Do we actually stratify patients, as to whether or not they have a good or a bad microbiome? Now, this is critical, preconditioning regimen. Now, do we wipe out the inherent microbiome before we give microbiome modulation strategy? And we'd use that if we give an FMT or anything less. We have to really think critically about this, and I'll show you some data as to why. How do we administer these different agents? And then during the therapy, what type of therapy do we combine this with? Immune checkpoint blockade, chemotherapy, other therapies, and then how do you assess the impact? So really a lot of considerations as you take these into account. Now, how about the role of diet? And Dr. Cromer really elegantly highlighted some of this, wanted to share some of the data that we generated on this. And given the critical role of the gut microbiome, what is the role of diet and other factors in response to cancer treatment? Okay, and so microbes in the gut are actually influenced by a number of different factors, including diet, antibiotics, and other environmental factors. Physical activity, sleep, right? All of these things matter. And numerous studies have actually shown that patients who receive treatment with antibiotics, broad-spectrum antibiotics, before treatment with immune checkpoint blockade, fail to respond to therapy. Okay, many different studies have now shown this. Not only do those patients have worse responses, but they have dramatically worse survival. And Laurent Vogel was one of the first people to show this. Really important work, suggesting that should we be really careful about antibiotic stewardship in patients undergoing treatment with immunotherapy and immune checkpoint blockade, but perhaps even with other forms of therapy. And in addition to this, I think, are there other factors? Can pollution actually contribute to changes in the gut microbiome and other aspects of disease? And I think absolutely yes. And Charlie Swanton just presented data at ESMO to suggest this. And these results have potential implications for patients with earlier stage disease and implications far beyond cancer, right? Could we be fueling our next really epidemics by actually giving antibiotics too frequently? So how about diet? So we and others studied the role of diet and other lifestyles, lifestyle factors on cancer treatment. And so Chris Spencer and Jen McQuaid and Carrie Daniel, in the context of when we were studying patients with melanoma going on to treatment with immune checkpoint blockade, asked the question, well, gosh, shouldn't we be looking at diet as well? And so in every patient who came in addition to collecting an oral and gut microbiome sample, we also administered a diet and lifestyle survey to them by REDCap, and looked at the data, and we were blown away by this. What we found is that if patients reported sufficient dietary fiber intake of at least 20 grams per day, which I can tell you less than a third of patients actually reported that, they were 5 times more likely to respond to treatment with immunotherapy. And it's not just a threshold, it's more is more, right? And so for every 5 gram of fiber per day increase, there was a 30% reduction in the risk of relapse. Really profound. And so published these findings on Christmas Eve last year. So in addition to this, not only did we study this in our patient cohort, we also studied this in a preclinical model with Giorgio Trinchieri and showed that if you give mice a low-fiber diet, they fail to respond to treatment with immune checkpoint blockade, and they fail to activate T-cell signaling pathways. And also worked with others worldwide, including with Romina Goldsmith at the NCI, who showed that gut microbes and dietary fiber can actually shape the tumor microenvironment, in part by monocyte reprogramming by STING-mediated interferon signaling. You can essentially take a pro-tumorigenic tumor microenvironment and make it an anti-tumorigenic by feeding mice fiber or giving them gut microbiome modulation, or both. And so now what we're doing at MD Anderson Cancer Center is really acting on this. And so Jen McQuaid and Carrie Daniel and others are doing brilliant work to test diet as a precision intervention in cancer, with the hypothesis that a whole foods-based fiber-rich diet will actually modulate the microbiome and enhance systemic and anti-tumor immunity. We actually have a bionutrition research core where we have chefs. They actually package meals, and we'll, like Blue Apron, and we'll actually ship them anywhere in the country, hopefully anywhere in the world someday, and actually have engineered diets up to 50 grams of fiber per day, and are testing those along with ketogenic and Mediterranean diet and other diets. And we're seeing some really encouraging results. So this is a patient that Jen McQuaid and I treated. 43-year-old female, came in with metastatic melanoma, multiple sites of disease. Actually, you can see on the PET scan here, had this pretty large lesion. She started on treatment with immune checkpoint blockade and also went on to one of these diets and actually had a complete regression of her tumors. I actually am beaming, as is she, underneath this mask. Nonetheless, took out both sites of disease and found that there was absolutely no viable tumor. And so she's very likely cured of her melanoma based on this intervention. And so what does 50 grams of fiber look like in real life? And so this is from this book on cooking for your gut health. And I have this chart actually on my refrigerator that my kids and I use, to try and push our fiber up. In addition to intermittent fasting, we advocate a high fiber diet. But, you can see that some of these high quality foods like lentils, black beans, limas, chickpeas, chia seeds, but apples, it's 4 grams of fiber. With the skin, you got to eat a whole lot of apples. One a day ain't going to do it, right? So, but being intentional about fiber intake and really being able to advise our patients, I think, is critical. And ultimately, I think we need to embrace a more holistic approach to cancer treatment, which integrates monitoring and modulating of the microbiome, diet, and other factors alongside things that we're already doing. Sure, we're looking at, the tumor genomics. We're looking at systemic immunity-ish, right? But should we actually be profiling the microbiome and then stratifying patients into those with an unfavorable microbiome, a targetable one, or, an adequate microbiome in which we can actually use a rather extreme type of treatment like a fecal transplant or a targeted microbial intervention, or just support them with diet? And should we be integrating other host factors like sleep, stress, exercise, all these things? And, again, I revisit the question, could microbiome targeting become the next pillar of cancer care? And what I can tell you is we're getting there, right? So believe it or not, they've actually developed these smart toilets where you can actually sit down, it'll read your fingerprint, and actually profile your microbiome real time, send it back to your smartphone and give you automatic input on what you should be eating and other factors. There's 3D printed pills you can swallow, which will profile your microbiome all along the length and the alimentary tract. And then they have breathalyzers for disease, and they even have personalized nutrition from an interventional standpoint. And so with that, I know we're a bit over, so I will close. And if we have time for questions, but we may not, probably not. Okay, but thank you very much for your attention.
Thank you so much, Dr. Wargo. Oh, thank you so much, Dr. Wargo, and to all the presenters today. We unfortunately don't have any more time for questions, but if the speakers can perhaps hang on the side for a little bit, you can approach them with the questions. That concludes our session today. Thank you again to everyone for attending.